Good morning, everyone, and welcome to the 7th in the series of webinars from the Industrial Transformation Research Hub in Biocatalysis for Pharma. My name is Elizabeth Gillam, and I have the pleasure of coordinating and introducing these webinars. Before we start, I just wanted to mention a little bit about the Research Hub and these webinars. The Hub is a group of academic researchers with complementary expertise, which we plan to apply to problems associated with the use of enzymes as biocatalysts in pharma. We hope to engage partners from industry to help us leverage governmental and other funding to support our research on projects that are relevant to and designed with pharma. And I'm happy to say that we have currently engaged 2 companies, and we're significantly along the way to reaching our funding target. The emphasis in these webinars is on presenting some of the science that's led to the formation of the Research Hub. And through the webinars, we aim to introduce you to some of the members of the Hub and to demonstrate the expertise that they bring, as well as to illustrate some of the projects that we could pursue in the Hub. These webinars will also serve as a model, though, for what to expect from the regular forums in the Hub that we will use to communicate outcomes to ensure participants have timely access to information and to tools resulting from the research underway in the Hub. We're almost at the end of the series now, but if you've missed any webinars to date, we've got a YouTube channel to which all of the webinar recordings have been uploaded. Our speaker today is Dr. James Behrendorff, who is a middle career researcher in the Hub from the Queensland University of Technology. James comes originally from New Zealand, but did his PhD in Australia in the area of protein engineering, and specifically engineering of P450 enzymes for biocatalysis. After his PhD, James worked in industry for several years, and recently he's developed his skills in synthetic biology and now leads a research program that's focused on production of small molecules for industrial partners, combining approaches from the very small to pilot scale, which is what he'll talk about today. Thanks, James.
Thanks very much for the introduction, Liz. So in my research group, we use biology to make small molecules, but we don't do a whole lot of wholesale metabolic engineering in service of this goal. When working even with highly engineered microbial cells, there are a lot of biochemicals for which the maximum yield will always be limited due to metabolic pathway length, intensity of cofactors required, and poor compatibility with host cell metabolism. While the idea of making anything from simple sugars or even from CO2, if you have an autotroph, while that idea is very attractive, we believe that there are often better routes to achieve maximized yield and selectivity of a desired product if we are not constrained by whole cell metabolisms. Our strategy is to identify the simplest possible route to produce a desired product from any feasible low-cost substrate. Or the motto that we have in my group is to make anything from anything. I'd like to take you through a couple of examples of products that we're working on in my group. I can't identify the specific molecules as these are all from industry projects, but I think you'll be able to appreciate the workflows and types of enzymes that we're dealing with. These are compounds where there are good reasons for developing biocatalytic approaches. For example, some of them, their natural concentrations in the organisms that produce them in nature, their concentrations are typically at low micrograms per gram of biomass or lower. So there's a good imperative for pursuing biosynthetic methods. The first compound here is a flavone that is produced by a very constrained metabolic pathway. The preprint displayed on the right of the slide here is the last paper to come from Amyris, where they determined the maximum yields for over 240 small molecules produced in Saccharomyces or E. coli. And despite generating hundreds of thousands of microbial strains, several precursors to our desired flavone compound had very low yields and were among the worst performing products, even with Amyris applying their best AI-guided high-throughput strain engineering. So instead of trying to make this molecule from sugar and a yeast, we tried to identify the lowest cost feasible precursor. And for us, feasible precursor also means something that we can actually source and process locally. And we discovered that we could actually dramatically shorten the pathway to only 2 reactions. The first step in our workflow is characterization of the candidate enzymes. Some of you will have joined last week and heard the introduction to cell-free protein expression systems. But for those who missed it, this is a system that we use as a tool for rapidly expressing hundreds or even thousands of proteins in only a couple of hours in extremely low volumes. And generally, these proteins are functional and sufficient for activity assays. The first step in our new pathway for our product is an acetinosyl methionine-dependent methyltransferase. And this assay format is great for methyltransferases. It's compatible with a highly sensitive commercial assay that's available for detecting formation of acetinosyl homocysteine. And it is also highly amenable to other colorimetric or fluorometric assays for coenzyme A, for example, if we're looking at a CoA transferase alkylation reactions. And due to the low reaction volume, the cost of expensive substrates and cofactors becomes a non-issue, even when screening extremely large libraries. The second step in our new pathway is catalyzed by a prenyltransferase, and we are exploring options for high throughput screening of prenyltransferase based on the release of pyrophosphate. But in this case, it was really important that we screen for regioselectivity early in the process. So we actually went directly from cell-free protein expression into turnover assays and screened qualitatively for product formation and regioselectivity with UPLCMS, which provides sufficient sensitivity for these low-volume screening approaches. And although it obviously substantially decreases the possible throughput, we were still able to implement a 5-minute UPLC method that gave adequate information on selectivity to move forward quickly. After identifying the preferred enzymes for these 2 steps, we can continue working in cell-free expression initially to optimize the reaction conditions, including pH, temperature, buffer components, etc. It allows us to quickly discover which steps in a pathway are going to be compatible, and which steps are optimal in dramatically different conditions that might need separation, and where we need to find compromises. And then with an eye on further process development, we transferred the best candidates into whole cells for further testing. Whole cell biocatalysis is attractive in this instance because growing cells are required for SAM regeneration. We found that our substrate readily crosses the membrane in E. coli. So long as the cells are actively growing, they are quite rapidly methylated and the product readily diffuses out again. So the first step was almost too easy to believe, so the challenge is coming in the next step. The prenyltransferase proved more challenging, and there are strong reasons for wanting to express a prenyltransferase in whole cells, mainly the provision of prenyl diphosphate co-substrates from natural cellular metabolism. However, we found that our preferred prenyltransferase could not compete with E. coli metabolism for prenyl substrates, even though the enzyme works very well for us in vitro, and we get adequately efficient turnover. It just can't compete in vivo. But in vitro biocatalysis, supplying prenyl diphosphates directly to a purified enzyme is not really feasible due to the high cost and poor stability of synthetic prenyl diphosphates. So we had to broaden our scope a little bit, and we implemented a 3-step in vitro cascade for prenyl diphosphate supply from low-cost prenyl alcohols. Using this pathway shown on the bottom left of the slide there, we can start with just 5-carbon alcohols, and through the promiscuity of a couple of kinases that don't do these reactions in nature, we can arrive at the same universal 5-carbon prenyl diphosphate intermediates and produce any prenylated compound from these alcohols. And this compared to providing synthetic prenyl diphosphates, this is at least a 5,000-fold reduction in cost of goods. And the conversion of our intermediate substrate to the final prenylated product is really quite efficient with this system. So how do we knit these two things together? This is really the next stage of development for us. We've got a whole cell system methylating the initial substrate, and we need these cells to be actively growing to provide the acetinosyl methionine. And then we've got an in vitro stage for providing the prenylation step. And so whether we take this as a two-step process where we take the intermediate from the first whole cell incubation and then have a second step there, or whether we find a way to just separate these steps with the dialysis membrane, which I think might be possible, or whether we bite the bullet and go for an all in vitro process. Our methyltransferase that we're working with works very well and is very stable when it's purified. It's only a question of whether the cost of purchasing acetinosyl methionine at large scale at wholesale prices is worthwhile or not, how that weighs up against running fermentations to otherwise provide the SAM regeneration. We had a couple of bonuses come out of this project during the initial screening phase, and this is stuff that we wouldn't have been able to pick up if we hadn't taken this high throughput screening approach in cell-free. We made some discoveries by combining enzymes and compounds that are not normally co-located in nature, and discovered previously unreported enzyme promiscuity that allowed us to make some new to nature compounds. And these compounds were kind of exciting because they have the same functional group on them that's responsible for the therapeutic effect, but distinct properties elsewhere on the molecule that should result in different absorption, distribution, and excretion. So we've potentially got a pathway to developing alternative versions of the compound that we were initially interested in, but that have different therapeutic application. So to further inform this hypothesis, and it is just a hypothesis at this stage, we've undertaken an in silico docking screen of our new molecules against the entire human proteome, comparing potential binding sites of our new molecules to the original target molecule, and to understand whether they're actually likely to hit all the same targets in the human body. And this is in partnership with the Australian government's industry research organisation, CSIRO, who provide both the computational biology and the computing power for this part of the project. I'll go on to another example here. In the second example, we have a 5-step pathway for a natural product. Again, this is an industry project. It was a bit fun to be working on a longer pathway in this case, and we were able to implement the entire pathway in cell-free protein expression and do one-pot synthesis of the final product. Obviously, sensitivity starts to become an issue doing that many steps in very small volume, but we were still able to detect the product in LCMS. And so that meant we were actually able to look at combinations of different enzymes and optimize whole pathway cascades in the cell-free system. This particular molecule we're after requires a kinase, a SAM-dependent methyltransferase that actually catalyzes 2 steps, a P450 system, and a PLP-dependent decarboxylase. And in the process of trying to combinatorially optimize the pathway, we figured out we were actually just better off without the first step altogether, even though the first substrate going into the pathway is the lowest cost starting point. That first enzymatic reaction, which is catalyzed in this case by the PLP-dependent decarboxylase, was so slow relative to the subsequent steps. It was such a bottleneck that we're actually better off paying a little bit more to begin at the first intermediate step and actually eliminate one of those steps altogether. This also allowed us to do some work with P450s in cell-free. And this particular P450 was a nice case study because we know that its native form does not function in E. coli, and we weren't the only group to have tried and failed to express it in E. coli. Direct collaborators and more second and third degree connections who had spent a couple of years trying to work with this one in E. coli. It works very well in Leishmania cell-free expression. And so we were able to quickly screen a larger number of mutants and kind of some of the typical variants, but also some more, more combinations than people had otherwise screened, cloning things one at a time and checking them in E. coli. So we're just able to more quickly move through some options. And as I mentioned, the native sequence does work in Leishmania cell-free expression. And so we screened for mutants that were just better, where we got better activity out of it. And we're working with the assumption that it wasn't due to better catalytic properties, but maybe an improvement in folding stability, meaning a greater proportion of functional enzyme. And there was an assumption at that stage, but sure enough, when we took our best performer across to E. coli, it worked fine. And at this stage, the P450 dependent step is not the rate limiting step in the pathway, which is quite satisfying in the context of some of the skepticism that persists around using P450s in small molecule synthesis. One more point before I move on to the next slide. Another advantage of this system is we can combinatorially screen P450 and reductase combinations. And the end point of this investigation was that indeed the native partner reductase of this enzyme supported much better activity than any of the alternatives that we worked with, but we're able to show this combinatorial screening of P450s and reductases can produce quite meaningful and interesting insights. So again, moving from cell-free protein expression into something more scalable. And in this project, we've gone with the approach of having 3 independent E. coli strains for catalyzing each of the steps. So we're starting off with a substrate A going through a P450 reductase system to give us intermediate B. And it's nice to have this in the whole cells, of course, for the NADPH regeneration. Then we've got substrate B, diffuses out of the first cell and into the second, or rather it's transported, but transport is not a limiting factor. And that's phosphorylated by a kinase. And again, having that in whole cells is great for the ATP regeneration. And that intermediate also readily leaves the cell and gets into the third catalyst here that does the methyltransferase reactions. But we've got a problem here. We've got intermediate C coming into the cell with hosting the methyltransferase that needs to do 2 sequential methylations. And the second methylation is much slower than the first, and that intermediate singly methylated compound can diffuse out of the cell but not get back in. So we've got a problem. We've got a lot of loss of the intermediate into the supernatant. And we've got around this for now actually just by permeabilizing that final cell and going with biting the bullet and feeding with S-adenosyl methionine. So for now, we've got a functional process to go all the way from our selected substrate to the fully methylated final product and at wholesale prices versus the value of this particular compound. It all still stacks up economically, but this is a really interesting opportunity to find a better methyltransferase for the second methylation step. In nature, it's a single methyltransferase doing both steps, but I suspect we could pan a much greater variety of methyltransferases and even find independent enzymes for doing each step. So these biocatalysts are prepared in fermentation that we can grow them up individually to prepare bulk massive cells and then store them for later use. Another thing that we like about having them in, let's say, each catalyst in a separate bag is it's easy to titrate in different concentrations of each of the enzymatic steps. We can obviously characterize the activity per cell unit for each of these, and then we can figure out, okay, should we have 5 units of this cell and 1 of this cell, and balance the pathway for optimal flux. Because we grow up a lot of biomass for this part of the project, we do routinely dry down in different formats, lyophilized or we've got a range of drying technologies, and we also characterize the stability of the catalyst in those dried formats, and we are actually able to dry and repeatedly reuse this P450 reductase system. It's a good segue into our other piloting facilities here at QUT. We've got good traditional integration with agriculture and with fermentation, and so we've got we're well equipped for being able to move from molecular scale to large scale with our projects. Our biocommodities pilot plant is located about 1,000 kilometers from Brisbane, and the reason for that is it's co-located with a biorefining pilot plant directly attached to a sugar mill. This is sugar growing country up in Mackay, and so there's effectively no shipping cost on the feedstock to do piloting work there. One of the aims of the pilot plant is to basically have duplicated capabilities between our city campus and the pilot plant, but at different scales, and get us in as good a position as possible for being able to develop processes at our city campus that can be translated efficiently to pilot scale. So on campus, we have fermentation from 1 to 30 liters, and at the pilot plant, we go from 100 to 2,000 liters. And these piloting scales really complement well the capabilities of our hub partner, John Woodley at DTU. The separation technologies are also similarly transposed, I suppose, to pilot scale. So on campus, we can do continuous cell disruption at about 6 liters per hour, and also tangential flow filtration and lab scale chromatography for separation. And then at the pilot plant, that goes up by a factor of 100. The drying technologies are also quite good here. I believe we're the only university in Australia that has spray drying in a PC2, a physical containment level 2 lab. So we can do reasonably highly engineered atypical strains in spray drying here, and that's quite exciting for us. We also have the usual freeze drying and rotary evaporation technologies. And then at the pilot plant, we just have all the same, but much bigger. And the microwave assisted vacuum drying is the scaled up equivalent of a rotavap, effectively, in terms of the conditions that the sample is exposed to. We also have some direct experience in my group with trying to stabilize very complex biological materials. So we've developed a lyophilized version of the cell-free protein expression extract that we use. It's the first example of a eukaryotic cell-free expression extract that's been successfully stabilized like this. It's more than 40 essential biological components, so we have to find conditions that can accommodate all of these components and their storage and subsequent revival. There's also more to cell-free expression than just the cell lysate. These lysates are always topped up with a feeding solution of some kind, and for us that's amino acids, RNTPs, also another polymerase. And we had to really look into whether we could combine all of these things together or whether they need to be separated for lyophilization. And we worked through a reasonable matrix of different bioprotectants. and looking at whether we had everything pulled or separated out, and eventually settled on a satisfactory combination of live protectants. And going for on the slide here, the third possibility, where in our Behrendorff tube containing our reagent, we just have physically separated the cell-free protein expression lysate and the feeding solution prior to lyophilization. And we got a pretty satisfactory outcome out of it. So on the left-hand side here, we have freshly prepared cell-free protein expression extract, stored at different temperatures, and the y-axis here is showing the amount of protein that the system can synthesize when we wake it up again. We want to do protein expression. You can see that at elevated temperatures, the activity is lost rapidly. At 37 degrees, in reality, it's probably gone to zero within the first day. And it's storing at 4 degrees. This is kind of what we expect. There's a decay and capacity for protein synthesis. And about half of it is lost in 2 weeks, which is, although we can do a lot with the system, we can express all sorts of proteins and detect their activity quite readily in these very small volumes. If you're suddenly halving the expression capability of the system and this is using GFP as a model, it's the easiest possible protein you can express. If you're losing half of it in 2 weeks, then this is, it's not possible to ship it out other than at -80, and that's how we do ship it to people around the world on dry ice. With the live protectants that we developed, we found that at 4 degrees, it was just as good at storage at -80. So shipping on wet ice is immediately within range, and we saw a little bit of decay with shipping at 24 degrees. So that's within range of packing it in polystyrene and sending it in a FedEx envelope. You obviously wouldn't want the shipment to get caught on the tarmac uninsulated in Singapore or Abu Dhabi, but this is a pretty good improvement already. And wet ice, or at least insulated shipping, is vastly more affordable than shipping on dry ice, obviously. And thank you again for your attendance today. And just to remind everyone that the talks are all on the YouTube channel here with the channel name Research Hub in Biocatalysis. Thank you, Liz. I'll hand back over to you.
Thanks, James. And with that, I will open the presentation up to questions from people online. I will, however, stop the recording now. To preserve confidentiality and anonymity of anyone who wants to ask any questions. So thank you to people who are listening in person and online. And feel free to, if you haven't managed to ask questions of James, feel free to send some questions through, and I'm sure he'd be very happy to answer them. Thanks very much.